Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here today with Graham Dempsey, who's the CEO and co-founder of Quiver Bioscience. They're at Cambridge, US. I'm in the original Cambridge here, but your Cambridge is very nice, too. They're a Cambridge-based biotech company that works on next-generation therapies across the central nervous system. So we're going to talk about antisense oligonucleotide programs and also Graham's experience more broadly on AI-driven drug discovery, which is obviously in growing importance today. So, Graham, thanks for joining us.
Yeah, thank you for having me. Excited.
Let's start with Quiver. What drove you to co-found the company? And if you could tell everybody a little bit about what you're working on, let's start there.
Sure. So the motivation behind Quiver is really to address what have been some of the challenges in discovering medicines for the nervous system. It's incredibly hard. We like to say it's creating a new medicine is almost as hard as anything you can imagine. Then doing it for the brain is like harder than trying to land on Mars.
Yeah.
So it really requires, from our perspective, new technologies, new tools, new approaches. So Quiver was really founded to build a new approach to discovering new drugs for the nervous system. And we've really thought about it from where we see our important gaps in the way in which companies develop drugs. And you can think of it like, the brain is, first of all, as a tissue, is not easily accessible the way, a tumor is in cancer. So the ability to actually create model systems to study it with human context is incredibly hard. There's been a lot of very exciting work in developing better model systems using stem cell technology. So that's one important pillar of what we do, is creating models of the brain and different diseases, really focused on different types of neurons, because different neurons you have many different cell types in the body, so you want to be able to make the right cell type for the right disease. So that's a first pillar of what we do at Quiver. And then second is, how do we then derive important information about what happens in an actual disease? And you can think of it this way, which is your brain is inherently an electrical organ, and electricity, bioelectricity, and is really how you think, it's how you feel, it's how you act. And of course, it's what is fundamentally disrupted in many disorders of the nervous system. It turns out it's really hard to measure and to measure with cellular precision and at a scale that is amenable to drug discovery. So that's another aspect of this that we focused on is building instrumentation software that allows us to measure those signals, all using light. So we've invented ways to convert bioelectricity into light, which, as you know, is a way of scalably being able to measure these signals. And then lastly is the way in which we leverage AI and machine learning. You can imagine the scale of the data sets that we generate.
Yes.
Massive, because we can now measure across many millions of single neurons per day, per instrument. And really, it's beautifully fit for purpose data sets to extract fundamentally what are the patterns of bioelectricity that are altered in disease, and then how can we use that information to make decisions about creating better drugs.
And just to follow that thread exactly, how today, you talked about how hard it is to develop drugs for the brain. How are you using that Is it about identifying the right targets and something in the electrical signals points you to that? Is it about measuring if it's working, almost like a biomarker of
Right.
or is it a little bit of both? Maybe you can talk through where the biggest gaps are that technology closes.
I would say it's a bit of both. And we talk about our current programs that we're focused on, we start with genetically validated drug targets for those programs. And in that case, the platform is really built to help you select the best molecule, both from the perspective of are you reversing or changing the fundamental disease physiology, and also avoiding unwanted toxicities, which is a big challenge in really any space. So that's, I would say, for our current lead programs. But then to your point, what's really exciting to us is the ability to then use these for discovering new targets. And as you know, there's one of the challenges in the pharma industry is that there's a tendency to go after a lot of the same target biology. So it's like a small number of targets with most of the gravity focused on pulling resource. But my view is that, especially in neuroscience, we need to get to better target approaches for different disorders, and likely, as you know, polypharmacology and being able to modulate not just one target but multiple targets is likely going to be the longer term solution.
So that first point you made was really interesting to me because it closed a little bit of a loop in my mind, which, if I've got it right, it sounds like you can really use the electrical signal that you can measure to get a picture that is preclinical, allows you to optimize the molecule, figure out even if you're on the right, if it's a genetically validated target, you're starting quite a ways down the road, but still you really want to be able to show before you go into the very expensive and time-consuming human trials that you can correct that electrical phenotype in a model, right? So is that the right way to think about it?
That's exactly right. Exactly right.
So I think maybe we start with the pain program that you're working on. It's a really interesting target. Maybe you could start by just telling people about this target, and then I'd love to maybe just dive into that as an example of what's the phenotype we're measuring, and how do you think about going about to correct it?
Yeah, it's a very exciting time to be working in the pain space, and there was a very important drug approval earlier this year from Vertex in January, a drug called Journavx, or suzetrigine, which is a small molecule targeting an ion channel called NaV1.8. So certainly very exciting, mostly impactful in the acute pain space. Chronic neuropathic pain, I think, remains a bit elusive in terms of getting to very effective non-opioid, non-addictive based medicines. So the story and the target that we're focused on is NaV1.7, and that is one of the other channels that is expressed in your sensory neurons, and your sensory neurons are critical for transmitting pain signals from your periphery to your brain. And turns out that the genetics of this target are quite interesting, and it was identified many years ago that there were patients that were identified that had loss of function mutations in this gene for NaV1.7, and they do not experience pain.
Right.
Congenital insensitivity to pain, which, is problematic.
It's not good. Yeah.
Evolution has put these mechanisms in place for a very important reason, so it's quite problematic. But in any case, it's very, very compelling. And then it was also identified that there are gain of function mutations in this channel that cause extreme forms of pain. So as a drug developer, as you know, when you're looking for genetic validation of a target, this is, I would say, as compelling as you could possibly imagine, short of the mutation in the gene causing the disease exactly. So now this, of course, set off a very exciting race in the pharmaceutical industry to develop small molecules, because you could imagine now, if I can develop a pharmaceutical agent that can control or turn down NaV1.7, I should be able to control pain
Yeah.
while keeping nociception. That's the sort of the critical, because you still want to be able to experience, you still want to have those sensations and feelings.
Sorry, what can you define nociception for those who don't know?
Yeah. So, pain is, threshold above, what would be your normal perception of your environment. So you don't want to lose the ability to feel that you're touching something warm.
Yes.
But of course, you want to be able to tune down if you are experiencing pain.
Yeah.
Now, it turns out that developing a small molecule for this channel has been elusive, and many companies have tried, and really all have failed so far. And the reasons are many, and it's important to keep in mind that there's important learning that's happened over the last 20 years in the field. And interestingly, small molecules and the challenges that developers have faced have been, one, getting enough of the drug to the target to block it with sufficiently to be able to control pain. So that's one. Two is selectivity. Now, it turns out there's a whole class of these ion channels, the SCN gene. You have NaV1.5, which is expressed in your heart. You have other sodium channels in your brain. So ultimately, you want to be able to control that one without modulating the others. And that, of course, turned out to be pretty challenging. And groups have figured that out, but it's been difficult. And then, I would say most recently, one group in particular in phase 1 trials, they identified on-target toxicity with the small molecule related to what's called hypotension. So patients basically fainting when they take this small molecule drug. And it's really because it turns out there's a little bit of this channel that's expressed in the part of your nervous system that controls your heart rate.
Oh, wow.
Yeah. So now our approach to this is we've taken, I think, a very different approach, which is a genetic medicine or an antisense oligonucleotide. And in that case, our approach is to deliver the antisense directly to the tissue through a lumbar puncture. And this is how all of the neuro-approved drugs in the ASO space have been approved. So this is deliver it directly to where the tissue is. We can engineer in selectivity, and we can avoid those toxicities. And that's really our approach. And I think it's very compelling from a genetics perspective. It's just been very difficult to drug with classical small molecules.
And if we think about the patient population that this is most targeted towards, can you talk a little bit about the scale, the size, the unmet need there, just to help people understand? Because obviously a lumbar puncture is only accessible for, but could be potentially life-changing for a certain segment of the population. Yeah,
great question. Where we're starting are really severe forms of chronic neuropathic pain. So, and that includes indications like small fiber neuropathy, metastatic bone pain, which is related to cancer pain, diabetic peripheral neuropathy. So when you start to look across indications, it becomes quite impactful for millions of potential patients ultimately, even though it's a lumbar puncture. What we like to, when we think about this as a delivery vehicle, millions of epidurals are given every year.
Yep.
And frankly, it's about procedurally, it's approximately the same. So I would say that to start, we're going to focus on moderate to severe forms of chronic pain. And then my sense is that as we improve some of the chemistries, we improve some of the deliveries, then that window will be able to open more broadly, but quite impactful, we think.
You have another program, I understand, in developmental disorders. I did all my PhD work in rare and ultra rare developmental disorders. So I'm really curious to talk about that side as well. I think also this points to the platform that you have, right? You can go after chronic pain on one side, but also again a neurological condition, but very different. And after we go here, we can talk a little bit about the opportunities and challenges that come from having this this platform approach. But it's great if you could talk a little bit about the other programs in your pipeline as well.
Yeah, happy to do that. And the platform itself, as I mentioned, one of the things that's very unique to what we do is the scale with which we can measure neurophysiology. And if you think about pain and epilepsy, those are clearly disorders of excitability, of alterations in excitability of the relevant neurons. And so it's very front and center for us. That was part of our thinking was, okay, these are highly aligned with what we are very unique in world at doing. So exactly as you said, neurodevelopmental disorders, and we have a program in a disorder called Dup15q syndrome. And in that particular case, you're dealing with a duplication, and the critical gene is UBE3A, which is a ubiquitin ligase. You may have heard of this gene in the context of Angelman syndrome. Mm-hmm. And now in that case, patients are missing expression of this protein in their brains. And drug developers, including some of the leaders in the field of ASO drug development, like Ionis and Ultragenyx and others, have come up with ways to turn on the silenced copy, because there are two copies, and turns out that one of them is just naturally silenced in the brain, and there's a way of actually developing an ASO to unsilence that gene. So to silence the silencer, so to speak, that allows that to turn back on. And those trials are actually in later stage development, I believe in phase 3, and expecting readouts in the first half of next year. And the anecdotes are amazing. I mean, I think we don't appreciate how plastic the brain really is ultimately. And there are certainly reports of patients that are nonverbal prior to receiving this medicine, actually being able to verbalize. And if you're a parent,
that's amazing.
It is amazing, right? Just being able to hear that your child is in pain or needs something, or to hear them say that they love you, things that you take for granted as a parent. I am a parent, so pretty profound. And I mention that only because I think it shows, first of all, it has direct relevance to the gene that we're interested in, and certainly shows that this modality has the potential to be transformative and disease modifying, ultimately to be able to not only address symptoms, but actually start to try to address some of the underlying issues with the disease.
And my understanding is Dup15q is relatively common amongst the rare and ultra rare developmental disorders, right?
Exactly, it is, and it's you can think of it on par with Angelman syndrome, actually. And in the case of Dup15, our objective is to, because there's too much of it, is to knock it down. And in that case, it's highly amenable to some of the different approaches that have been developed for using antisense oligonucleotides for controlling gene expression.
Yes. This is maybe a good lead into the double-edged sword of being a platform company. You can go after, especially RNA and oligos are a platform, the electrophysiology and understanding the brain is a platform. So you've got a huge search space, which is a great thing, but it also means you've really got to focus. I'd love to hear a little bit about how you break, upon which axes are you choosing to focus now, and how do you think about that?
Yeah, I think it is a great framing, because there are lots of options that we have, but we're a small company, so we've got to make some very important choices to begin with. So I think for our lead programs, in many ways we've tried to de-risk along the target dimension, meaning that we're picking targets that we know have very strong genetic validation, and therefore, and as you know, drugs that are developed against genetically validated targets are are quantitatively more likely to get towards approval
Yes.
and impact in patients. So I think, de-risking along that dimension, I think is very rational to start. And then in that case, the platform is used for qualifying and identifying the best molecule, the best performing molecule. And that's where we can use the platform, as you were alluding to at the very beginning, Patrick, about creating patient-based models of these diseases, being able to, especially where mouse models in particular are have currently limited translatability, we can really lean into those models to help us understand how electrophysiology is altered in patient derived cells, and then use our approach to then find the molecule that is best, both at reversing those disease phenotypes and avoiding off-target or as we say, off-vector toxicities, perturbations to neurons that you would want to avoid. So that's how we start, and as I mentioned, epilepsy and pain, I think, are areas of tremendous need, and again, align very well with some of the things that our platform is very unique in world. So that's why we've started in the space that we have. That being said, I will tell you that we have this. One of the things we're just very excited about is we have the potential to impact many diseases, and that's really what we want to be able to do. And I'm certainly happy to talk about how we think about the breadth of opportunity, and certainly start to prioritize what are our next programs that we want to take on.
Yeah, I want to get your perspective on that in the field more broadly. Before we jump into that, I want to cover a little bit of your background, because you spent a ton of time understanding super resolution imaging, diving deep into understanding what's going on in the brain and other tissues. Maybe before we talk about, let's talk about that. But I also just want to understand, like, if we think about something like pain and a stem cell, how can I conceptualize what pain looks like in a stem cell? Like, is there a characteristic electrical signature? How are you measuring that? And maybe you can use that to just talk a little bit about the world of understanding what's going on at a cellular level.
Yeah, sure. And I might go in a few different orders here, but certainly what does pain look like in a dish, in a stem cell or stem cell derived cell? Well, one of the, I think, amazing things that's happened in the space over the last many years is being able to harness the knowledge we have of developmental biology to understand how to make different types of neurons. And there's been a lot of learning, even more so recently with all of the transcriptomic technologies that are now pervasive, to understand how do we coax a stem cell to have the characteristics of a sensory neuron, for example. So there's ways to do that, and we've been developing our own methodologies, generally focused on transcription factors that we know are important in pushing a stem cell towards that particular fate. So take it that we can do that in the lab, we can do it very well, reproducibly, and make billions of these types of neurons. And what you want to do is then make sure that those neurons express the targets that are expressed in the body, so that's important, and that the cells respond to pharmacology in the way that you'd expect them to respond. So there's ways to do that, to build confidence about the nature of the cell that you're making. So assume that works, and that you can get to a sensory sensory-like neuron that you can make in a 384 well plate. Now you ask, what does pain look like in a dish? And ultimately, the way that I would say a lot of people have thought about this is very binary, which is things go up, things go down. So cells are either more excitable or they're less excitable. I think we could do a lot better than that. And that's one of the things we're focused on is how do we measure the full breadth of alterations of the physiology of those neurons? Generally speaking, the neurons become hyper excitable in a disease state, whether you induce that through an inflammatory stimuli, or as we do, we can actually make a patient mutation associated with small fiber neuropathy and show that excitability is altered. Yes. So you do see changes in the excitability itself. So cells become hyperexcitable, but a lot of aspects of that are different depending on how you stimulate the cells to fire. So this is one of the things we do here is you can think of it like we put the neuron through its paces. We can stimulate the cells with very brief, very long stimuli, strong, weak, and then we look at the full range of response of those neurons across that range of stimuli. That gives you a very rich set of features that define the response of that neuron, a few hundred electrophysiological features. And this is where the machine learning tools are particularly powerful to say, okay, what is the set of physiological features that are altered in, let's say, a genetic model of small fiber neuropathy, where we've introduced a gain of function mutation into NaV 1.7. And what does that mean? What is that distance relative to your healthy basal firing? So it's in many ways we're putting it into this map, this high dimensional map. Now you have a vector, you have a target that you're trying to get your drug to push. Exactly.
Interesting. Yeah.
Right. And what's very interesting, when a lot of times when you think about oligonucleotides, you think, oh, okay, well, as long as I, at the molecular level, change that target, then all ASOs are effectively created equal.
Right.
And I can tell you that is unequivocally that is wrong. So, and when you start to look at molecules that effectively knock the target down to similar degrees, so molecularly they're acting similar, functionally they can be very different, meaning that some are just inherently more perturbative to the functional response of the individual neurons.
Do you have any idea why that is? Yeah, what's driving that?
Well, it is an area of very strong interest in the oligonucleotide field, because for a lot of the way that it's done, development is done in the space now, you have to empirically determine and identify these sorts of CNS associated toxicities, doing very high dose studies in animals, mouse and non-human primates, and often these sort of longer term toxicities, you find them too late in the process. Now you've done a very expensive set of studies and you say, oh, geez, okay, now this is a problem. Well, it turns out that there's a variety of ways in which oligonucleotides can interact with neurons such that they can be acutely perturbative in terms of how they interact with the cell itself. And then over longer time frames, it's likely that these molecules can interact with other RNA binding proteins, start to alter some of the response of those neurons in ways that you obviously don't want them to. So there's quite a bit of interest of thinking about the mechanistic toxicities. I'd say it's not fully understood, but empirically you can start to measure and train on these learnings, right? Where you can start to learn the rules of design and chemistry so that you can say, okay, these are aspects of the design that I want to avoid, and these are ones that we know are going to be clean in terms of their response.
Yeah. It would be interesting if you could take all of the data from those high dose animal studies and then cross correlate that with the stem cells, right? To build that model. And I'm sure you're doing that, but yes. It could be an interesting industry-wide collaboration, right? Because a lot of people have this data.
It is broadly interesting to the entire industry. And as you said, this is where I think AI and machine learning is particularly powerful, is there's lots of data that already exists where you can start to understand these relationships between design and toxicities, for example. And exactly as you said, we're generating some of these training sets specifically now here at Quiver to build our machine learning tools around predictivity. It definitely would be something that I would see as very broadly interesting to anybody that's developing an oligo-based drug.
Yeah. More broadly, what have you learned about working in a biotech company? It's a unique and leading and co-founding one, obviously. It's a unique situation where you have a really small team, especially in the beginning, and you've got so much to do. I'm just curious of your learnings from a leadership, company building perspective.
Yeah, I mean, it's awesome. I love it. I'll start with that. And of course, it definitely cycles between complete fear and total excitement. So I won't sugarcoat it in that way. It's very, very challenging, but it's super fun. And things that I've learned. Team is just everything. I get to talk to you, Patrick, about all this amazing work, but as you know, there's an awesome team underneath that is in the lab doing important work. So I think team sport, importance of building a great team and operating with efficiency and effectively is one thing that's really worth the time spending in terms of build. So I think that's something that's super important. And persistence, it's hard, right? You constantly have to make decisions, and you have to ultimately decide when things are worth pushing forward when they're not working.
Yes.
And I think we're often are in favor of shorter term gains, but if you study the companies that have been very successful, they often have had to push through very difficult, experiences to get there. And I think a level of persistence, or for me, it's perhaps pathological persistence. I think it really is important to get through to building something that's really important and special. So I think it's super exciting. It's hard. It's a lot of fun. But the environment we're in now, I think takes a lot of persistence to get through, to get through it.
Yeah. What talk about the environment right now. And I'd love to get your perspective on, the future, especially when it comes to genetic medicine, precision medicine. It's been a challenging year for biotechs in general. It seems to be like it's maybe getting better now, but I'd like your perspective on that because you work with a lot of companies. Obviously, you're out there in the field yourself as well.
Yeah, I mean, I'm inherently an optimistic person, and I think that's somewhat required in the space. Is you got to believe that you can get through it. And my sense is that it has been a difficult year, but there has been a lot of excitement and a lot of progress in a number of different areas. So there are new medicines that are being made. Perhaps the most obvious is the success in the GLP space, where these medicines really work. They really work incredibly well. So it does demonstrate what's possible with persistence, because that field has been around for quite some time, and now has traction. So I think the innovation engine continues to turn. I think that you see real successes in the space. That's just one to highlight, but I think there are many more to highlight. And then oligonucleotides in particular. We mentioned a few particular areas that are also looking really exciting in the epilepsy space, in our development space, in particular. So there are drugs that are showing real promise and showing that they can work. So I remain optimistic. I think my sense as well is that as we get through the turn of the year, there's been a lot of churn at many levels of our world this year.
Yes.
I remain optimistic that those will become less so, and more opportunities are going to open up. Opportunities both for interacting with pharmaceutical companies and also on investment as well.
What is the most interesting technology area for you right now that isn't directly related to the work that you're doing at Quiver? Is there something that you look at and think it's really exciting, even though if it's not something you have the resources to go after right now yourself?
That's a really interesting question. And I would say that certainly in the oligonucleotide space, I talk about oligonucleotides broadly, but there are actually quite a few subclasses of different types of molecules that so siRNAs, antisense oligonucleotides, obviously gene editing technologies that are around. We don't have the bandwidth to do all of these things. But I would say that the field of oligonucleotides incredibly exciting in terms of the progress that's being made. And I think there's also a lot of progress on ways to deliver those molecules across the blood-brain barrier. So there are exciting companies that are building shuttling technologies, for example, to get, such that, because as you may know, if you want to get an ASO into the brain, you have to inject it intrathecally.
Yeah.
So certainly, and talking about this in the context of pain, certainly as we come up with ways to get those molecules to cross the blood-brain barrier such they can be delivered peripherally, I think that is super exciting, and also to the extent that they can be targeted to specific brain regions and areas of the nervous system. So not only crossing, but being able to get to the desired region, I think is really quite exciting. And some of those I believe will start to become ready for prime time. Not something that we're working on, but something we're quite interested in, and a lot of exciting progress.
Can you explain a little bit about how those shuttles actually work? How do they do their magic to get in and cross blood-brain barrier and go to the right place?
Yeah, and I won't attempt to speak for all of them because I know there's a variety of ways, but often leveraging transferrin receptors, so receptors that are known to enable internalization and therefore accessibility to the brain is one, I think, relatively common way that folks are deploying these tools. You can think of them in the context of antibodies that can carry a payload of some molecule that allows it to cross the BBB, the blood-brain barrier. So that's certainly one canonical way. I think that's pretty well known. But certainly there are, I think, other mechanisms and approaches that companies are trying to exploit to enable that approach.
Adding to my list of rabbit holes to go down to learn more. It is, it's something that I know conceptually, but I couldn't tell you about all the nitty gritty roots of how they do that. But it strikes me like it sounds like it strikes you as one of those things that if we can crack it, it takes all the magic of RNA medicine that we're seeing everywhere else and opens a whole new organ system.
Yeah, and the other one I would quickly add is combination approaches as well. So it's often that we think about one target, one drug target, and therefore it has a massive effect physiologically. But I think work that people are doing to rationally approach polypharmacology, a lot of drugs that work very well often are hitting not just one target, but multiple targets. And it's not easy, though, to rationally design in that way. But groups that have been able to figure out some very compelling approaches where there is a good understanding of those targets can then actually leverage the biology of not just one target, but multiple targets. So I think that is really something we're very excited about in some of our approaches, and even thinking about pain, thinking about ways to modulate not only NaV1.7 but also NaV1.8. So taking a dual strategy is something that we see is really exciting for the future.
So how do they work in going after them at the same time? What do you get that you don't get by just one or the other in isolation?
Well, we don't know yet for sure, but when you start to look in our platform, actually, it's quite interesting because you can see that each of those channels contributes differently to the excitability of a neuron. Right. So one is most important in driving when you're very weakly stimulating the cells. One becomes greatest in terms of contribution when you have very strong stimulation to the cells. So they have differential contributions to how action potentials are generated in those cells. So if you really want to shut down action potential firing, being able to modulate both gives you the strongest potential effect. But it's, as you can imagine, non-trivial in terms of getting a drug-like molecule or molecules that can do both.
Yeah, interesting.
Yeah.
I want to finish off on maybe a fun story here. My understanding is when you were starting your career, you actually had dreams of being a medical doctor, sports physician. I'm curious what was your sport of interest, and where did you go astray that you ended up doing RNA medicine?
Yeah, it's as they say, it only makes sense when you look backwards in terms of connecting the dots of your life. And yeah, I started, as you said, I originally aspired to be a medical doctor. I thought I would be on the sidelines for the New York Giants, which is my football team here in the U.S. They're not very good this year, but in any case, I'm a huge fan, and I thought I would be the physician on the sidelines, interacting with the players, and that was kind of my original ambition. And it really was after I started in college and started to get into doing real research. And I said, okay, I want to actually try this. This is really, really interesting and exciting. And I was at the University of Pennsylvania in Philadelphia. I didn't have really experience, and I sent around, cold emailed, I don't know how many professors to try to get an opportunity to join a lab to do some sort of research.
Yes.
I just wanted to try something. And I heard back from one lab, and ended up at his name is Yale Goldman, at the Penn Muscle Institute, doing single molecule biophysics. And it was amazing because Yale he's a clinician by training, but also an electrical engineer by training. So as I would joke, he would be running a class on cardiology and then be in the lab fixing a EMCCD camera himself. I was amazed by that, and that was what really got me, just by good fortune, kind of got me the bug of being excited about biophysics and technology. And that's, everything came out of that. That's where I ended up going to Harvard and doing my PhD in the biophysics program at Harvard Medical School. I worked with Xiaowei Zhuang, as you mentioned at the start, developing super resolution technology. So it's something that it's certainly a theme in my career and my life, is developing really powerful, novel platforms for biology. And super resolution was amazing. It was, you may know that some of our competitors won the Nobel Prize in chemistry,
right,
for this discovery. But in any case, it was amazing to contribute to that, and really shows the power of optical technologies. You really look at what drives genomic sequencing technologies, what drives the recent spatial transcriptomics technologies, optogenetics, high resolution microscopy. It's all light-based, really novel light-based systems for doing these very, very high resolution measurements. And that theme is now carried on to my work here at Quiver in terms of our work in neuroscience.
You, I said that was the last story, but actually there's another one from your background that I thought was really interesting, which was, my understanding is I think it was at Penn, where the program was funded by Roy Vagelos, and I think you had a chance to meet him. So Roy was CEO and president of Merck, also chairman at Regeneron, is really a legend of drug development. It sounds like one of these kind of examples where you probably don't know who you're in the room with at the time, and then years later you're like, "What? Why didn't I take that opportunity to get a job or something?" Maybe you could tell that story.
Yeah, well, it's funny because first of all, he is certainly one of my heroes in this space, and all the work that was done there at Merck. And I didn't necessarily foresee myself getting into biotech and biopharma and drug development. It just wasn't necessarily in my immediate view at the time. And but I was, as part of my time at Penn, I was biochemistry and biophysics focus, and Roy very generously through the Vagelos program funds individual undergraduate students for their education at Penn.
Cool.
And I was one of them, and I got a chance to meet him and his wife, and definitely did not appreciate the magnitude of that at the time. But again, as I think it was Steve Jobs said, right, and perhaps in his when he gave his commencement speech at Stanford, I think it was it's only when you look backwards that you say, okay, now I see how this all comes together. Looking forward, it's really hard to do that. That strikes me as a moment that was really impactful for me, and certainly the folks at Regeneron and Copolis and all those are certainly heroes of mine and their work.
Well, Graham, this was an amazing conversation. I'm super excited about what you're doing as a company. I learned a ton. Thank you for spending the time with us and sharing all this and all the great work you're doing.
Thank you, Patrick. It was an absolute pleasure, and I really appreciate you having me on.
Thank you, everyone, as always, for listening, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate it if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.